Hi, my name is Rainbow Desilva. I'm a recent graduate from the Department of Environmental Science, Policy and Management at UC Berkeley. I'm going to talk today about the association of genetic and climate variability in giant sequoia. So I want to provide a really quick overview. So first, we're going to talk a little bit about giant sequoia ecology. Then we're going to go into the details of this study of local adaptation in giant sequoia. Then we're going to touch on the expected changes to Sierra Nevada water balance, as it's particularly relevant to giant sequoia. Then we'll talk a little bit about other resilience indicators in this species and what these lines of evidence might help determine as to the future of giant sequoia. And then we're going to talk about management options. What can we do as managers to facilitate population persistence in this species? So giant sequoia is a long-lived tree species. It occurs in 70 to 75 groves across mid-elevation Sierra Nevada forests. And as you can see in this figure on the right here, which shows giant sequoia distribution in purple overlaid on April snowpack, this distribution is highly fragmented. We have a fire-adapted species, so the foliage tends to start really high in the canopy, and this species has really thick bark, so it can withstand a lot of fire. Ecological studies to show that high sensitivity to water availability, water and nutrient availability in this species. So for our study, we wanted to capture the entire latitudinal range. So we sampled 18 populations, including the northernmost and the southernmost populations. They're all labeled here. We genotyped 143 individuals using dd-RADseq. And after filtering, we obtained 1,364 biallelic SNPs. So we used a combination of FST outlier tests and genotype environment association to look for signals of local adaptation in this species. So we used two genotype environment association methods, latent factor mix models, which is a univariate method. That means that you're associating single locus, single predictors with each other, and redundancy analysis. And this is a multivariate method where you can combine suites of environmental predictors to predict your allele frequencies, for example. And on all the indicated SNPs, we completed a blast search to get an idea of their functional significance. So we found 79 SNPs as outliers. So 6 of these were FST outliers, and here the FST values range between 0.55 and 0.72. So these are quite high FST values. Our LFMM suggested 58 outliers. RDA, which is our redundancy analysis, we found 24 outliers through RDA. And 9 SNPs were indicated by these two methods. Now, of these 79 SNPs that were outliers, 19 of them had functional annotations when we blasted them, so to speak. So our results kind of show that this signal of local adaptation in this species is really dominated by precipitation-related variables. So in this top graph, what we see is that PC1. So here in our latent factor mix models, we use composite variables. We did a PCA to transform our variables. PC1 was driven almost entirely by temperature, and PC2 was driven almost entirely by precipitation-related variables. So here we find that PC1, we have 7 outliers, whereas PC2, we have 51 outliers. So we're seeing a much more strong associations between precipitation-related variables and SNPs. And this story was very similar for the RDA. So we completed model selection for the RDA with our full suite of environmental predictors. So this told us that the three environmental predictors with the most predictive value were precipitation of the driest quarter, which for Mediterranean ecosystems is really summer precipitation. Climate water deficit, which is an indicator of drought that's very important for Mediterranean climates, and isothermality, and that's temperature fluctuation, to kind of put it in a nutshell. But for all of the outliers that we found with the RDA, they were all most correlated to precipitation of the driest quarter, and the next was climate water deficit. So this is showing us that drought or moisture availability is really important as a selective agent in this species. And giant sequoia is likely have high sensitivity to water availability. So if you think about that, if you think that we're saying that giant sequoia is locally adapted to gradients and moisture availability, in Sierra ecosystems, snowmelt offers an important source of water, especially in the spring and early summer, where we don't have any rain typically, or very little rain, I should say. And snowpack in the Sierras is rapidly declining, and it's predicted to continue declining by up to 70% by the end of the century. So less snow equals a longer, drier season. So a longer period of time where seedlings that might be particularly sensitive to drying out have to endure each year to survive to the next year. So just to kind of wrap things up a little bit. So giant sequoia as a moisture sensitive species whose natural distribution is becoming increasingly arid. So we might have locally adapted populations that are going to be maladapted to the future. And so Aiken's group at University of British Columbia came up with this framework where you can adapt, migrate, or perish if you're a tree population. So if we kind of think about those options for giant sequoia, we can think about the adapt. So the positive here is that we found a signal of local adaptation, meaning some populations are likely more drought tolerant than others. So that's good. But when we looked at neutral diversity in giant sequoia, we found across the range neutral diversity is quite low compared to a lot of other conifer species. I like to add adjust as well because there's a certain amount of phenotypic plasticity. I think it's relevant for giant sequoia because recently we had a drought and over 100 million trees died in the Sierras. And giant sequoia didn't suffer a lot of mortality, although there was some foliage dieback. So I like to say that as like, we saw a phenotypically plastic response in giant sequoia. But physiological traits can have tipping points. So just because we haven't seen the mortality doesn't mean that we won't. Doesn't mean that a small incremental change in water availability might be catastrophic for giant sequoia. Migrate. So we have found that the likelihood of migration outside of range boundaries is likely quite low for giant sequoia. So it has very long generation times, approximately 300 years, and modest dispersal distances. So this is going to limit the range at which the species can track climate changes by migrating out of range boundaries. So the individuals can't migrate. What about the genes? Can the genes move around? So we also find that gene flow is quite low among these fragmented distributions. So if there are drought adaptive genes, they likely won't move around on their own at a very fast rate. So this kind of brings us to the management. So unfortunately, the outlook might be a little bit bleak. Like, we don't have a high likelihood of adaptation through natural variation or through the influx of adapted genes. But we can do things. We can reduce alternative threats. So climate change is ongoing, but there are other risks to giant sequoia. Currently, there's a huge accumulation of biomass in these forests that is creating these unnatural fires that are really hot, and we are seeing mortality where we never saw mortality before. And so we need to think about thinning and prescribed burning in giant sequoia forests. Extensive seed banking goes without saying, just collecting and conserving germplasm, and then also assisted gene flow. So if there are populations that are drought adapted, should we go there and sample these and spread those genes around? Or if there are populations, which there are, that have really low diversity, should we be planting to enhance the diversity? These are important questions that we need to think about and consider the context that some of these low diversity populations are also genetically distinct in composition. So we have to kind of weigh our pros and cons here, talking about the future. Lastly, I just want to touch on some of the people that were involved in this work with me. All right, thank you very much.
Hi everyone, my name is Jill Sekely. I'm a PhD candidate over in Germany, and my project is looking at local adaptation in an Andean tree species. And yes, I'm aware there's quite a bit of irony in being an American who's employed by the Germans to go to Argentina to study a tree species and figure out how it lives its entire life in one place. But hopefully I'll be able to learn something from this experience and actually apply it to my own life. So the overall point of my project is studying local adaptation in situ, and I'm sure many of you are familiar with how difficult this actually is to do in the field, just because there's so many things going into local adaptation and they're so difficult to actually pick apart. So my project looks at all the things on this screen, but for time purposes, I'm only going to focus on the things listed in black. So the three things that you mostly have going into local adaptation are genotype, environment, and the phenotype. For genotype information, I use SNP data. I use this to look at the population genomics. For environment, I use the CHELSA dataset, which is very similar to WorldClim. And then I use these two pieces of information and I will do environmental association analysis. I've just started doing this, so I don't have any results yet, but I will share some results from the other two parts of the study. So one of the ways we can make this a little bit easier on ourselves is to choose a nifty study system that has pieces that are a little easier to pick apart. And for this study, we went down to the Andes Mountains in the southern tip of South America. The cool thing about the Andes here is that they run almost perfectly north to south, which creates a couple of very clear environmental clines. The first, as you can see from space, is a very clear rain shadow, so precipitation gradient as you move from west to east. So you have a lot of rain over on this side, but then you get into the steppe once you move east. The other thing is the temperature and day length gradient associated with latitude. So I'd like you to remember to flip your brain upside down here and remember that in this case, north is actually hot and it gets colder as you move south down into the ice fields and down into your Tierra del Fuego. The other thing we need is a good study organism. So we went with Nothofagus pumilio. This is related to the Fagacia family up in the northern hemisphere, so beach. And this species has a very long continuous range from 35 degrees south, so basically subtropical, all the way down, continuously running into Tierra del Fuego. So that's over 2,000 kilometers of unbroken forest. It's a very high altitude and cold adapted species, and it often forms natural monospecific stands up at the tree line. It can grow up to 1,500 meters above sea level at the northernmost parts. But the other question you have to ask is how exactly do you get good information out of this without breaking your bank, without breaking my brain? And so we went with a paired study design after Lauterhaus and Wheatlock 2015. And their idea here is that you choose pairs of sites scattered across the landscape. And the idea of a pair is that they're close enough that they share an evolutionary history. However, they're far enough apart that they are under different environmental selection pressures. So the idea there being that you can then kind of ignore the neutral demography or it just doesn't exist. So here's an example from one of the southern sites. These two pairs are, they're less than 2 kilometers apart. However, there are 300 meters in elevational difference. So there's a very strong temperature difference between the two. We also did 3 unpaired extreme sites over at the edge of the distribution. So this is a xeric site. This is kind of a very cold and high site, and then we have a very warm and dry site up here. We have a total of 500 adult trees. And to give you an idea of what this actually looks like on the ground, here are some of our study sites. So this is the xeric site out in the steppe, just a couple of trees barely clinging to life. And then you have kind of a more normal looking forest. This is down south, and this is all Nothofagus pumilio as far as the eye can see. So down here in the valley floor, up into the hills, and then all the way up into the tree line. And morphologically, there's also a lot of interesting things happening. So you have kind of your normal tree looking trees, and then you also have some kind of Middle Earth looking trees as well. For the environment data, we use CHELSA data set, as I said before. This is very similar to WorldClim, where they basically took real data and then extrapolated across the entire world. And this is good news for South America because there's really not that much climate data available otherwise. So the CHELSA data set it's 19 bioclims, and essentially these are mostly related to temperature and precipitation. Some are just regular variables, some are kind of synthetic. But to look at how this actually translates into my sampling sites, I did a PCA analysis. So along the bottom line here, we have PC1, which is mostly temperature variables, explains 56% of the variation. Along PC2, we have mostly precipitation, 23% of the variation. And then these colors, if they're similar color but different saturation, that indicates that it's a pair. So this is a pair, this is a pair, etc. And you can see right off the bat that even though these sites are very close and none of them are more than 2 kilometers apart from each other, they do occupy quite different environmental niches, which is good news because that's exactly what we were going for. For the genetic side of things, we started with a list of candidate genes and essentially did targeted sequencing. So our candidate genes came from a couple of different sources. We have some colleagues down in Argentina working with this species for a long time. They had some ideas. We took some from a similar project that's happening in Europe right now called GenTree. We also have a transcriptome that was assembled de novo a couple of years ago by our colleagues for this exact species. And then using that information, I extracted DNA from leaf samples, and then we have about 50,000 SNPs that came out of that analysis. And so far what I've done with that is looked at the population genomics. So here is one of the major modes from admixture, looking at probably 4 genetic clusters. It might only be 3, but this is probably the most likely. And you'll notice one thing is that so the warm, dry site pretty much always breaks off by itself. There's definitely something weird happening there. But otherwise, they kind of line up just by the geography. So there's probably like a north cluster, a central cluster, and a south cluster. And then I'm going to also use this information to look at the demographic history. So like many montane areas across the world, there's been glaciation patterns that have caused bottlenecks and expansions and all this other things. And so I'm going to start looking into that next. I also looked at isolation by distance, so essentially a mantle test. And each one of these points is the pairwise genetic distance between 2 of the populations. And so the blue are not paired, the red are paired, so these are our paired sampling sites. Along the bottom, we have the distance in kilometers, just a linear distance. And then over here, we have the genetic distance as determined by FST. And a couple of things that pop out right away is that our sites, as expected and as hoped, they are not very far apart genetically. The other thing is these kind of outliers are all from that weird northern population. And so downstream analyses, I think we're probably going to cut those out, or at least compare the data set with that population and without. And then the other thing to notice is this kind of general pattern happening down here. So up until you get about 750 kilometers away, it's probably gene flow that's influencing the genetic differentiation. But then as you get further away, it might start going more into the drift. So that'll be some good information for later on. But the important thing to note here is that we did get that exactly what we were going for. Again, like with the environmental data, the sampling site strategy did work out in the end. So hopefully we'll be able to take all of this information and going into downstream analyses. So the next step, as I mentioned before, we have the demographic history inference, which I'll do with the FASIMCOL, possibly also stairway plot, which is non-model. And then I'll start doing the environmental association. So actually looking at the SNP outliers that I've already found, but I haven't yet associated, but judging by how the environmental distance and the genetic distances are stacking up so far, it looks like we will have some pretty interesting results. So please stay tuned and follow us for more updates. Thanks.
Hello everybody, my name's Ian MacLachlan. I'm a postdoctoral research associate at Texas A&M University. Today I'm going to talk about effect sizes and multilocus genotypes, and whether they can be used to estimate climate-related phenotypes in lodgepole pine. This work is an extension of my PhD research at UBC with Sally Aiken, whose lab has been focused on polygenic adaptation in climate-related traits for a long time. We recently published a study that showed across natural and selected seedlings from 12 breeding zones, we can summarize the frequency of alleles associated with climate-related traits, both in relation to their phenotypes and also in relation to their climates of origin. Now, each point on this plot is a breeding zone mean genotype and mean phenotype. And behind each genotype, there are approximately between 10 and 30,000 allele counts summarized. And I've been curious for a while now as to whether these breeding zone level analyses can be implemented at the individual seedling level. So I have 2 questions. Can individual phenotypes of adaptive polygenic traits in lodgepole pine be explained by multilocus genotypes? And are SNP effect sizes useful for characterizing phenotypic variation? SNP effect sizes are an essential component of polygenic risk score analysis that's been developed in human genetics over the last 10 to 15 years, and is now coming online as a major clinical tool to estimate phenotypic risk from genotypic data, but I'm not aware that it's really been applied in plants, let alone forestry. So genotype-phenotype associations identify the effects of an increasing or decreasing number of alleles at a given locus. Each GPA effect size is the slope of the line, be it positive or negative, depending on which allele is the reference allele. It is the allele that has the positive effect on the phenotype we define as the positive effect allele. And so, for each locus, we can identify the positive effect allele, and we can calculate how many of those alleles each individual has at each locus. If we take those scores and then multiply them by the effect size, then we obtain seedling PEA score, a polygenic score associated with that seedling and that set of loci. Now, using the previous data for lodgepole pine in 105 natural seed lots and 20 selectively bred seed lots, I calculated PEA scores. I used genotype-phenotype associations with natural seedlings for height and cold injury traits. And by calculating the polygenic scores for each seedling, I can show that PEA scores for height have a clear relationship and explain a moderate to large proportion of variation in height phenotypes. PEA scores for cold injury also explain quite a large proportion of variation in the cold injury phenotypes of the natural seedlings. However, the panel of SNPs identified from the GPA analysis of natural seedling does not closely match the panel of SNPs that is adaptive in the selective seedlings from the same region to accurately describe the variation that they contain. Now, the original plots that got me interested in this were based on positive effect allele frequencies. So I went back, and for each individual seedling, I calculated the PEA proportions, summing the number of positive effect alleles across loci for each individual. And I've called these PEA proportions which are within individuals rather than frequencies to avoid confusion with frequency-based population level statistics. Calculating these PEA proportions simplifies the analysis by eliminating the effect size component. And from these PEA proportions, using genotype-phenotype associations of natural seedlings, I can explain more variation than it did using PEA scores alone. And so these are strong relationships, and they are potentially useful in the future. However, like the PEA scores, they don't translate well to the equivalent selected seedling populations. So thus far, both PEA scores and PEA proportions can explain substantial amounts of variation in phenotypes, and PEA proportions explain more variation in all cases, and in cold injury that variation is quite substantially larger. This is likely due to error in the effect size estimates and the fact that PEA proportions are summarized over a large number of loci. Now, using the PEA proportions, I can also associate them with climate and show that the relationships between PEA proportions and climate are moderately strong, and again, stronger for cold injury than they are for height. It probably reflects the fact that cold injury is the strongest adaptive trait we know of in this species, in this part of the range. Now, to flip this around and use selected seedling genotype-phenotype associations, we also find that the selected seedling genotype-phenotype associations, positive effect allele proportions can explain large amounts of variation in height and in cold injury. These clear relationships reflect again the conservation of resistance within and among breeding populations as an important adaptive trait. But again, the genotype-phenotype associations do not identify standing genetic variation well in the natural populations, and so do not estimate and do not explain variation in the natural populations very well. Now, using these selected seedling GPAs and calculating PEA proportions, they're also very strongly related for both height and cold injury, both very strongly related with mean annual temperature, which is interesting and potentially useful if you want to manage adaptation in relation to mean annual temperature. So, in conclusion, using genotype sampling a range covering 1,000 kilometers and 6 degrees C in mean annual temperature variation from 12 breeding zones and one that includes some introgression from jack pine, it's possible to explain moderate to large amounts of variation in height and cold injury using multilocus genetic data. Now, this works for both the polygenic score approach and the PEA proportion approach. But with the data available, phenotypic and climatic relationships with PEA proportions are stronger and simpler to calculate. The results I found also suggest that you can't translate results from genotype-phenotype associations in natural seedlings to those of selected seedlings, or vice versa from selected seedlings to natural seedlings. In relation to managing climate adaptation, we can now envisage a seed transfer risk in terms of molecular data. Now, the seed transfer risk envisaged by Bob Campbell in the 1980s can be related to the mean annual temperature declines with cold injury PEA proportions, for example. And for a given mean annual temperature optimum of a population, you can determine the genotype frequency and the shift in that genotype frequency, which has more recently been defined as the risk of non-adaptedness in a purely molecular context. In addition, from a breeding program perspective, simplified management of genomic variation in polygenic adaptive traits in multiple tree breeding programs seems to be possible, and there may be predictive potential for growth and adaptation as long as predictions are within the limits of the populations used to determine associations. Limits in terms of climate, geography, and whether seed lots are natural or selected are far greater than the current limits of genomic selection approaches. And lastly, refinements and validations are required here. I have some technical refinements I want to do, but most of this data, in fact all of it, comes from a single seedling common garden. A much larger multi-environment study would be needed to identify markers and generate robust predictive relationships between PEA proportions and phenotypes and PEA proportions and climate in order to guide forest management decisions based on individual seedlings. And with that, I'd like to acknowledge everybody who's helped me out with this work, especially Sally Aitken for her continued support, as well as Lauren and Brandon at UBC recently, and Sam and Tegan at the University of Calgary, all of whom are my co-authors on the paper that started me thinking about this in the first place.